In this lecture we'll cover neurotransmitter receptors. The receptors come in two types: ionotropic receptors, which we'll cover in the first part. These are ion channels. The other option are metabotropic receptors. These are covered in the second part. These are also called G protein coupled receptors. Metabotropic receptors exert long-lived but slow effects on neuron membrane potential and potentially even gene expression. They take a little longer, and that's because they use G proteins to activate enzymes to stimulate metabolism within the cell, hence metabotropic. So we'll go over the general receptors, ionotropic, metabotropic, then we'll go through specific examples for individual neurotransmitters in the third part of this talk. So ionotropic receptors are simply ligand-gated ion channels. That is, once the ligand, the neurotransmitter, binds, the ion channel opens. There's no middleman, so we get a rapid change in membrane potential. Here we can see two different components for synaptic currents, or synaptic potentials. Notice there's an early hump with the E and a later hump with the L. They're varying the membrane potential, and this is just to show you the reversal potential for these two currents. And what we find is that the early component is mediated by chloride. And we know that because this reverses around minus 76 millivolts. That's shown in panel B. Notice the open circles cross the x-axis around minus 76. The later component reverses at about minus 102 millivolts. So while the early one uses chloride, the later component uses potassium. Both are inhibitory, and both, it turns out, are mediated by GABA. GABA will stimulate an ionotropic receptor for the early component, as well as a metabotropic receptor for the late component. So, the same neurotransmitter can create fast effects with an ion channel or slow effects with a G protein coupled receptor. We'll start with ionotropic receptors because they come first in the synaptic response, and they're also first in my slideshow. So, when you think ionotropic receptor, you should be thinking ion channel, and in particular, ligand-gated ion channel. These still have an ion pore, of course. You're not much of an ion channel without it. And they have a selectivity filter, meaning they let only certain ions move. Importantly, what we have here is a ligand-binding domain to serve as the activation gate. So when neurotransmitter binds, the activation gate moves a little and pulls the pore open. Kind of like what we saw with voltage-gated ion channels when we covered action potentials. Now, the ion channels aren't just floating around freely in the membrane. They are held in place with a large collection of scaffolding proteins. And collectively, this forms what we call the postsynaptic density. The postsynaptic density appears very, very dark in an electron micrograph because it's made of a whole bunch of proteins. So in order to be a functioning postsynaptic site, we need to, of course, have the receptors, these little jelly beans here. So these are ionotropic receptors. And to prevent them from moving around and potentially leaving the synapse, we have scaffolding proteins that bind to each other and the channels themselves. And of course, we need to eventually link this up with the cytoskeleton. In this case, it's going to be a whole bunch of actin. Now we've connected our surface receptor to the internal cytoskeleton, and that's going to provide a little bit of structure and prevent it from moving around. Now the receptors here are going to respond by opening up when ligands bind, generating an ion current. However, we should keep in mind that receptors desensitize. So we can think of this as the ion pore kind of relaxing. So when neurotransmitter binds, my pore is now open and ions can move through the membrane. After some period of time, and this depends on the receptor we're talking about, the pore is going to relax a little bit. There's our relaxation. So imagine that the ion channel is kind of like sucking in its gut when the neurotransmitter binds. But eventually you get tired of doing that and your gut sticks back out, and now ions move, but it's a little slower. There's just simply a little more resistance here because this receptor is desensitized, meaning it's not as sensitive to the neurotransmitter. Even though it's bound, we're going to get a much smaller response from this desensitized channel compared to the non-desensitized channel. Now, how fast is this? It could be essentially immediate. If we look over here on top, that's showing you a glutamate receptor called an AMPA receptor. And you'll notice it immediately desensitizes. In less than, I don't know, about 10 milliseconds, you basically get no more current. NMDA receptors, on the other hand, notice the change in time scale. Now we're looking at the course of ballpark, I don't know, 2.5, 3 seconds, and we're still getting a current. Yeah, it's cut down by about half, but we're still getting something. The AMPA receptor lets its gut go, and when it does, it completely blocks the channel, where we get no response once we're desensitized. So how quickly desensitization happens and the completeness of desensitization varies from channel to channel. But keep in mind, every channel desensitizes. Now, our ionotropic receptors can be either excitatory or inhibitory. Excitatory receptors increase the probability of firing an action potential. Inhibitory receptors decrease that probability. And they do this, as you probably guessed, by using different ions. The excitatory receptors are, for the most part, non-specific cation channels. That means they don't care. As long as you got a positive charge, you can move. This means not only does sodium enter, but potassium also exits the cell, as the cartoon is showing us on top. So that top left ion channel is a non-specific cation channel. Okay, so we got positive charge coming in, positive charge flowing out. What's the reversal? If I know the reversal, I know exactly what this ion channel is going to do. And the reversal potential is 0. That doesn't mean that nothing happens. 0 is quite depolarizing. It's well above the threshold potential of, like minus 55 or something like that. Now, on occasion, you do find some sodium channels. And those are very depolarizing, but also incredibly rare. If you had to bet, if you hear excitatory receptor, you should think non-specific cation channel. Whether you're non-specific cation channel or a sodium channel, either way you cause depolarization. And the whole point of doing that is to initiate an action potential that hopefully propagates on down the axon, brings in calcium at the presynaptic terminals, and we spit out neurotransmitter again. Inhibitory receptors, on the other hand, use chloride. Chloride still reverses at minus 76 millivolts or so, so this is going to hold us at rest. It might cause a little bit of hyperpolarization, but not much. Think about the driving force. A whole 6 millivolts of driving force. So you're not going to get much hyperpolarization, but what you will do is shunt the excitation. That is, remove the excitation. And that's what the bottom is showing us. So that EPSP, the excitatory postsynaptic potential, depolarizes membrane potential. If you look on the right plot, they have an EPSP and then an IPSP right after. And notice, we quickly lose the excitation. This is because when we are depolarized, that increases the driving force for chloride. So we really don't get much of a chloride current unless we try to excite. Think about this. If a neuron is resting, does inhibition have any effect? No. It's already resting. What do you want it to do? You can't do less than nothing. However, if you're trying to excite a neuron, that's when inhibition matters. We want to prevent action potentials from happening. We don't want to have even more nothing. That's impossible. So this illustrates shunting inhibition. Notice on top there they have a control plot that's showing us the excitatory postsynaptic potential without any inhibition. That's my EPSP. But if I get excitation with inhibition at the same time, this is my EPSP with that simultaneous inhibition. Look, we've lost all of this depolarization. We're missing out on all of that excitation. It's essentially been shunted away. That's why it's called shunting inhibition. And the product of this is shown on the bottom. On the bottom we see the firing rate on the y-axis and the amount of excitatory input on the x. Notice the difference between the gray control, that is without inhibition, and the kind of yellow, orangish with inhibition. By having the IPSPs, that inhibitory input, we decrease the firing rate. You'll notice we start firing pretty much immediately without inhibition. With inhibition, there's those 3 dots that stay at 0. That is, the neuron does not fire. That's what inhibition is all about. Not about hyperpolarizing an already silent neuron. So we're getting input at our synapses, and we're going to have multiple inputs for anything to happen because every synapse is incredibly weak in the central nervous system. So that means we're going to be summing the input from multiple synapses. Here's the thing. 1 plus 1 almost never equals 2 to a neuron. Now, what in the world does that mean? Okay, what it means is that if you get input from one synapse, and let's say the EPSP is, oh, I don't know, about that big. Let's call it a unit of 1. 1 millivolt, whatever you want to call it. If we stimulate synapse 2, and it also gives us 1 unit of depolarization, what if we stimulated both synapses together? Alright, so let's get us a little neuron over here, and we've got 2 synapses coming in. Synapse 1, synapse 2. Let's see, what would synapse 1 and 2 look like? Well, you would expect that we get 1, 2 units. That would be the expectation. That's not what we see. We see something less than 2 if the input is weak. Now, why is that? Why doesn't 1 plus 1 equal 2 anymore? Well, it's not really 1 plus 1. The depolarization from one synapse is going to weaken the input from the other. In other words, we're going to change driving force. Ooh, let's do some calculations. Okay, so if we're at rest, minus 70, and our reversal is, you got it, 0 millivolts. Well, 0 minus 70 equals, we have a whole 70 millivolts of driving force. Okay, we've now depolarized by some value. I don't know what. Let's just call it 1. We still reverse at 0, but we're no longer at minus 70. Ooh, we're at minus 69. So now we have 69 millivolts of driving force. Therefore, we will not expect the same current, or the same degree of depolarization. And that's what all these data over here are showing us. When you depolarize, so if you look on the top there, they have a nice dotted line for the reversal potential. When you depolarize, you get closer to reversal. Therefore, the driving force reduces. So subsequent currents are weaker. Therefore, 1 plus 1 equals less than 2. On the bottom, they're showing you a couple different simulations. So here, we'll just worry about synapses 1 and 2, because that's what they're putting together on the bottom left. So synapse 1 is blue, synapse 2 is an orange color, and what you'll notice is that they're about the same size. That gray line that they're showing, or it might be dotted, I'm getting old, that's the arithmetic sum. Okay, so let's just add it up. Just add these curves, you should get this. But what you actually get is the black line. So when they co-stimulate those two synapses at the same time, they don't get as big of a response. So the synaptic current isn't summing up linearly here. And part of that has to do with the change in driving force. Hey, let's not forget all that stuff about desensitization either. What if I stimulate the same synapse over time, rather than two different synapses at the same time? So let's give us two quick ones at synapse 1. So I get an input here for the first stimulation. If I stimulate again very quickly, I will not get another one because my receptor is going to be desensitized. So I'll get something a little smaller. 1 plus 1 will add up to barely more than 1. Now this might sound like synapses are always on the losing end. Not quite. There are occasions where 1 plus 1 is greater than 2. And that's when we're not dealing with weak inputs. Instead, we deal with strong inputs here. So what happens if we increase the strength? And now we're dealing with very strong synaptic input. Well, we're still going to have the same issue, where we depolarize and we're going to change driving force. This is absolutely true. But what we'll also do is change which receptors will give us a current. So let's say we have input here at synapse 1. And as I said, quite strong. Now let's imagine they're over here and with a little bit more detail. We'll just put them right next door to illustrate the point. Okay, so synapse 1 gets a little excitation and we depolarize. Well, that depolarization is going to spread rapidly all throughout the dendrites. That means the neighboring synapse will be depolarized. Now maybe we remember, maybe I will remind you right now, that NMDA receptors have that little magnesium block. And it keeps trying to come into the cell so long as the cell is very, very negative. Ooh, what if we've depolarized this synapse and we get rid of the magnesium block? Well, now we still have good old AMPA receptors. And in addition to stimulating those, we also stimulate NMDA receptors. Here's the deal. NMDA receptors, as we just looked at a couple slides ago, don't really desensitize. So they're going to move a massive amount of ions into the cell. Thus what we're going to get over here is not your expected amount of depolarization. No, no, no. It'll be much bigger. 1 plus 1 will be greater than 2. Because what we've done is increase essentially the number of receptors that are moving ions. So all this is just to say neurons do some crazy math. And it doesn't really work out like our math does. But they're always listening to multiple synapses. Sometimes the conversation at one synapse weakens the conversation at another. Other times, if there's a really good conversation going on, that can actually open up new topics at another synapse. In fact, that unmasking of NMDA receptors is probably very important for how we learn. More on that later, though. Okay, so ionotropic receptors, pretty simple, right? Ligand-gated ion channels, know the reversal potential, you know what it's going to do. Metabotropic receptors are a little more diverse. We're going to just cover 3 types of them, and they're going to be a lot slower. That's because they're going to use secondary messengers to create stronger and more long-lived effects on the cell. Okay, so metabotropic receptor, G-protein coupled receptor, it's the same thing. Now they're also exactly what they sound like, just like most things. A G-protein coupled receptor is a receptor that's coupled to a G-protein. How nice. What in the heck is a G-protein? A G-protein is a trimer, that is, it has 3 parts, that holds onto a G nucleotide, guanine, hence G-protein. So it's a protein holding onto a G. So there are 3 parts. There's an alpha subunit, that's shown over here, it's red, and then there's a beta and gamma subunit. Now, we usually put those together and just call them the beta-gamma complex. Beta and gamma stay in the membrane. So they'll move around a little bit, but the major player that we're going to talk about is the alpha subunit. And this is what makes each G protein different. It's the alpha subunit. When the alpha subunit is bound to GTP, as shown on top here, it dissociates from the beta-gamma complex and is free to roam around the cell. It will then stimulate some enzymes and cause some stuff to happen. More details to come. So for GDP bound, we're inactive. So we're going to float back to the surface, stick to our beta-gamma complex, and wait for another round of activation. So what causes that switch from GDP to GTP is binding to the receptor. So if the neurotransmitter binds, the receptor itself slaps a GTP on and gets rid of the GDP, activating the alpha subunit. What causes the transition from GTP to GDP is the fact that the alpha subunit is a GTPase. That means it turns itself off. So it loves GTP. It hugs it and squeezes it and eventually hydrolyzes one of those phosphate bonds there, turning it into GDP. And that cycle is shown on the bottom down there. We got our inactive GDP bound trimer on top, and then we got our active broken apart GTP bound alpha subunit. That alpha subunit is going to have really one of 2 targets. It'll either target adenylyl cyclase or phospholipase C. We'll go through these in detail as we need. So the G protein coupled receptors are responsible for that late current. And that's because there's just some additional steps. It's not neurotransmitter binds, ion channel opens. No, neurotransmitter binds, receptor puts GTP onto the G protein, alpha subunit dissociates, and then turns on an enzyme that makes a secondary messenger that activates a kinase, and maybe that kinase acts on an ion channel. Now really any of those bullet points over there work. Sometimes the G protein itself, like the beta-gamma complex, turns on an ion channel. And that's probably what's going on over here. Other times we make a secondary messenger, like cyclic AMP. And cyclic AMP then gates an ion channel. So there's several possibilities for how metabotropic receptors eventually turn on an ion channel. Now what's the whole point of this? Well, keep in mind at every step there's amplification. So one receptor will activate many G proteins. Each alpha subunit will activate many different enzymes. Each enzyme will make many different secondary messengers. Each secondary messenger can activate a couple different enzymes. So at each bullet point over there, we're amplifying our signal, essentially growing it exponentially, so we get a much larger effect, but it's slower. It's slower to come on, it's also slower to go away. So we can have pretty profound effects here using metabotropic receptors. Alrighty, let's get into some details. So the first target would be adenylyl cyclase. This is an enzyme that makes cyclic AMP. It's kind of like ATP, except there's only one phosphate, and that phosphate isn't hanging freely. It forms two bonds, so it's a cyclic nucleotide. That doesn't matter, but you should know cyclic AMP is a secondary messenger. Okay, so what's going on here? Let's get 1, 2, 3, 4, 5, 6, 7 transmembrane domains from Ig protein coupled receptor. And let's make this one GS coupled. That's my alpha subunit. I got a beta gamma over here. Truth be told, I don't really care about them for the purpose of this. They're wonderful, but we don't need to worry about their details right now. There, colored them in, that's the best I can do. The other flavor that we need to think about is GI. S, stimulate. I, inhibit. So GS is going to stimulate, GI is going to inhibit. I'm sorry, no chance of recovery there. Adenylyl or adenylate cyclase. Doesn't matter. Either one works. This enzyme catalyzes the reaction where we take ATP and turn it into cyclic AMP. Now we got our secondary messenger. So we got a G protein. If it's GS, it turns on adenylyl cyclase, increasing cyclic AMP. If it's GI, it turns off adenylyl cyclase, decreasing cyclic AMP levels. Cyclic AMP can then go up back to the membrane and activate something called a cyclic nucleotide gated cation channel, CNG. That is, it's gated by cyclic nucleotides, like cyclic AMP. And it's a cation channel. So that means positive charge is going to flow in. These reverse somewhere in the ballpark of 0, maybe -11, but definitely very depolarizing. So there we go. Now we can finally get a late current. That'll work. Hey, maybe you want to do a little more. Fair enough. Cyclic AMP's got you. It's going to activate a kinase called protein kinase A. Protein kinase A is then going to do whatever it does. It depends on the cell. I know it's going to add a phosphate group onto something else, but the devil's in the details. What type of neuron we're talking about, or what other type of cell, if you care about those, we just got to know. You got to look it up for that cell. What does protein kinase do to it? Does it make it more excitable or less excitable? Depends. Based on that, you can expect I'm not going to ask you, what does protein kinase A do in a neuron? Because there's no real answer. Maybe it makes it more excitable. Maybe it doesn't, right? Maybe it turns off voltage-gated sodium channels. It does that in some neurons. Hey, maybe it increases the expression of AMP receptors, making it more excitable. Hey, it does that in some neurons too. So just knowing that protein kinase A is activated doesn't tell us much, because every cell is going to treat protein kinase A a little differently. But no matter what the cell, protein kinase A will phosphorylate something, because it's a kinase. So a couple key words over there. They're bolded. Kinases add phosphate groups. Phosphatases remove phosphate groups. And keep in mind, phosphate groups have size, because they're made of atoms, and they have charge. They have a charge of -2. So they're going to change how proteins fold up, changing their structure, changing their function. Over here we can see the effect of protein kinase A on voltage-gated sodium channel currents. Notice, as you go from that top left to the top right current, it's getting smaller. And that's because it's been exposed to protein kinase A. If you like time plots, that's shown in B. The filled shapes are with protein kinase A, and the open circles are without. Now, of course, protein kinase A can also affect gene expression. So from here, the next important element that I want you to focus on, and hey, let's get the appropriate picture up, is CREB. the cyclic AMP response element binding protein. But we'll just call it CREB. What you need to know about CREB is that it's a transcription factor. That is, it's one of those things that will allow us to change gene expression. And here's where we get the most long-lived effects of metabotropic receptors. So if I'm GS coupled, I stimulate adenylyl cyclase, increase cyclic AMP, stimulate protein kinase A, stimulate CREB, and modify gene expression, allowing CREB to make the things that it makes. If I'm GI coupled, it's the opposite of all that. I will express less of those CREB-dependent genes. Okay, what are some CREB-dependent genes that we might care about? Well, what we're looking at over here in these data are is the expression of different glutamate receptors and their scaffolding proteins. Okay, so you look on the left, on the bottom. Here we're looking at, I believe, a Western blot, so it shows you the protein using an antibody. Control, normal mouse, nothing wrong with it. And then on the right, that CREB binding protein knockout. So they got rid of a protein that's critical for CREB to do its thing. So they prevented this, in other words. So on the right, we're looking at decreased CREB function. In other words, we're seeing what happens when CREB doesn't work. So GLUN1, GLUN2A, those are NMDA receptors. GLUA2, GLUA3, those are AMPA receptors. Subtle effects there, but the NMDA receptor effects are pretty obvious. When the bands aren't as dark, we're not making as many of them. So what kind of gene expression are we dealing with? Ionotropic glutamate receptors. These are really important. Bread and butter. Excitatory synapses. So if you want to strengthen a synapse, you need CREB. And in fact, when you knock out CREB, mice don't learn as well. The reason for that is that they're not able to strengthen their synapses in an activity-dependent manner. So there's our GS and our GI. The good news is we only have one more alpha subunit to talk about. That's GQ. Alright, so what's going on with GQ? We're not dealing with cyclic AMP anymore. We're dealing with calcium. That's our secondary messenger. So 1, 2, 4, 5, 6, 7 transmembrane domains. In this case, we're going to be GQ coupled. When neurotransmitter binds, we slap a GTP on and we activate the GQ subunit. We're not going to act on adenylyl cyclase. In this case, we're going to act on phospholipase C, PLC. What this will do is cut up some phospholipids, phosphatidylinositol bisphosphate, if you care, and it'll make two things. So we'll turn PIP2 into disoglycerol. That'll stay at the membrane. This will be important later, but we don't care about it for now. We do care about inositol triphosphate, IP3. That's what's going to stimulate calcium release. In order to do that, we're going to need ourselves a smooth endoplasmic reticulum over here, because on the smooth ER, we have these things called IP3 receptors. How nice! That means they bind IP3. So when you have an inositol triphosphate, it opens up these IP3 receptors. Hey, those are calcium channels that allow calcium to flow out of the smooth ER, so we get a little burst of calcium. But we don't really want a little burst. We want to send a nice, robust signal. And that's where ryanodine receptors come in. These are the same ryanodine receptors that we talked about when we covered muscles. I'm sure you remember everything from physiology. They're still calcium channels. So instead of getting a little bit of calcium to come out, we get a lot of bit of calcium to come out. That's what we want to see in a signaling pathway. We want big effects. So we get a burst of calcium because when a little bit of calcium leaks out from my IP3 receptors, it stimulates ryanodine receptors. And these are calcium-activated calcium channels. So it's not a little trickle of calcium. All of the calcium gets dumped out. Well, all, right? What does calcium do? It depends, okay? But it's going to activate this protein called calmodulin. And then calmodulin turns on a kinase. Cam kinase 2. Diacylglycerol and calcium collectively are going to stimulate protein kinase C. And just like protein kinase A, you got to know the details if you want to know what these do in a cell. So cam kinase 2 doesn't do the same thing in every cell. In other words, the devil's in the details. That's the good news. But we know they're kinases, so we know they're going to add some phosphates to something, and something's going to happen. And hey, guess what they act on? Creb. So the same story that we just had, we get back to. Because Creb is still going to change gene expression. So whether you're GS or GQ coupled, you can have long-lasting effects on a cell by modifying its gene expression through Creb. You get there through different enzymes and different secondary messengers and different kinases, but you still end up with Creb, and you can modify gene expression. So, let's go through each neurotransmitter, the receptors that they have, and now that we know what all these G protein coupled receptors do, we can figure out whether they're going to be excitatory or inhibitory. Let's start with glutamate, bread and butter. About 81% of the synapses in the cortex are glutamatergic. For the fast response, we're going to, of course, use ionotropic glutamate receptors. They come in 3 flavors. We never talk about the third, so we'll just talk about 2. We have AMPA receptors. That's your fast, postsynaptic excitation. That's what it's there for. These are not blocked by magnesium, so they're always available to work unless they've just worked recently because they rapidly desensitize. NMDA receptors, on the other hand, don't really desensitize. Both of them are nonspecific cation channels. Now, some AMPA receptors exclude calcium. So NMDA receptors are a little more calcium permeable, and they basically don't desensitize on the time scale that synapses work at. So you get a large burst of calcium from NMDA receptors. From AMPA receptors, you get very brief depolarization. That's it. So we have another source to get calcium. You don't necessarily need a metabotropic receptor to affect gene expression. You can also use NMDA receptors. These things are pretty nifty. So they're kind of like ionotropic and also kind of like metabotropic receptors because they bring so much calcium into the cell. And they're also kind of like voltage-gated ion channels, as well as being ligand-gated ion channels. And that's what allows them to detect coincident input. That means 2 inputs at the same time. That's why 1 + 1 is greater than 2 NMDA receptors. Okay, have a look at this IV plot over here. So here we're looking at the current on the y-axis. They're varying voltage on the x-axis. AMPA receptors give you exactly what you'd expect. A nice linear IV curve. Now these are both non-specific cation channels, so they're going to reverse around 0 millivolts. Of course, that varies a bit from cell to cell. But if I'm at the reversal, I get no current. If I'm above it, well, I need to get back down here, so I'll get an outward current. If I'm below it, I'll bring a positive charge in, giving me an inward current. NMDA receptors have a very similar curve until we get into the negative potentials. They have this J-shaped curve, and that's because of the magnesium block. At very negative potentials, the NMDA receptor is blocked by magnesium. It's only when we start to creep into these more positive potentials that we kick out the magnesium block and allow charge to move through NMDA receptors. So in order for glutamate to generate an NMDA current, I have to already be depolarized. So I must have already had some form of input. That's why we say it detects coincident input, when two inputs coincide. If you can get NMDA receptors to generate a current, you get massive calcium influx. Notice over here, the size of that AMPA component is very small for the EPSP, or in this case, sorry, it's a current, EPSC. The NMDA component is much larger. Look at the area under the curve. You're going to get a lot more calcium into the cell through NMDA receptors. That calcium is going to do what it did with our G protein signaling earlier. We also have metabotropic glutamate receptors. They come in all the flavors. There's, I don't know, like 8 different groups or something like that. Some of them are excitatory, like good old group 1, MGLURS. These are either GQ or GS coupled, depending on whether it's MGLUR1 or 5, but both of those are excitatory. GQ increases the calcium, that's clearly excitatory. GS increases cyclic AMP, you can get cyclic nucleotide gated cation channels, that's going to be excitatory. We tend to find these on postsynaptic sites. So again, glutamate's still excitatory. Group 2 and 3 are both inhibitory because they use GI or GO. They're exactly the same thing. Either GI or GO coupled GPCRs. So I don't know why GO is there. It's not different at all. Both GI and GO inhibit adenylyl cyclase. So I usually just say GI. Group 2 is found presynaptically. Group 3, postsynaptic. So depending on what MGLURs you have, glutamate could actually inhibit you. Now those group 2 MGLURs we think are probably more autoreceptors, where in case you're spitting out too much glutamate, you inhibit the axon, which, hell, it's not up here. So if this neuron is highly active, spitting out a whole bunch of glutamate, then it can stimulate this GI-coupled MGLUR, and that will inhibit that neuron, preventing further release, because we've already spat enough out. Enough with the glutamate. Speaking of enough with the glutamate, let's talk about some inhibitory receptors. So GABA, glycine, pretty much the same. I'm not aware of any metabotropic glycine receptors, but there are probably some. GABA is more commonly used in the forebrain, glycine, brainstem, spinal cord. But both of these have chloride channels. So ionotropic GABA-A receptors are just chloride channels. They'll hold us near rest and prevent neurons from being active. So GABA will stimulate these, but hey, so will ethanol. And that's what these data are showing us here. So we got baseline on the top, and then top middle, they had a little ethanol. Notice, slower frequency of action potentials. We've inhibited the neuron by stimulating GABA receptors. Wash out the ethanol, and we're back. Picrotoxin inhibits GABA receptors. So notice on the bottom, that's all with picrotoxin. Notice the firing rate's faster, and ethanol has no effect. There are other targets, benzodiazepines, barbiturates. All of them act on GABA-A receptors, stimulating them and bringing neurons closer to rest because they're using chloride channels. The same thing is true for glycine. Different distribution, different drugs act on it. GABA-B receptors are metabotropic. So here, everything's inhibitory. GABA-A gives you a chloride current. GABA-B, those are GI coupled. Those are going to decrease the activity of cyclic nucleotide gated cation channels. They are also going to directly activate potassium channels with the beta gamma subunit. So they're still inhibitory. That's all we need to know about GABA. GABA A, ionotropic, chloride. GABA B, metabotropic. It's going to decrease calcium and increase potassium permeability. Acetylcholine has two broad types of receptors. There's the ionotropic nicotinic receptors, and then there's the metabotropic muscarinic receptors. So nicotinic, ionotropic, and these are all cation channels. You find these at the neuromuscular junction, so we can depolarize muscle, move our body. You also find them in the brain. Typically they have a presynaptic location to facilitate depolarization of the presynaptic terminal, and thus neurotransmitter release. Largely what's used, though, for cholinergic signaling in the central nervous system are the muscarinic receptors. Now these are also used in the autonomic nervous system. And there are two broad types. There's the odds and the evens. The odds, M1, M3, and M5, are excitatory because they use GQ. M2 and M4 are usually inhibitory because they're GI, unless we're talking about smooth muscle. Remember, a decrease in cyclic AMP is going to actually stimulate smooth muscle, as we discussed in physiology. Dopamine receptors are either excitatory or inhibitory, but they are all metabotropic. There is no ionotropic dopamine receptor. There are two classes. There's the D1 class, which includes D1 and D5. These dopamine receptors in the D1 class are excitatory because they use GS receptors. The D2 class, D2, D3, and D4, are GI coupled and therefore inhibitory. Notice what's going on here. Here we're looking at the activity of a dopamine neuron. If we bathe on dopamine, the dopamine neuron pauses. That's what's shown on top. So dopamine turns off dopamine neurons. That makes sense. If there's a bunch of dopamine floating around, dopamine neurons probably don't need to fire. Let them rest. On the bottom, they're applying, it looks like quinprol. Quinprol stimulates the D2 class dopamine receptors. And notice there's a very long pause in this dopamine neuron. What's going on? Cyclic nucleotide gated cation channels. Just that simple. If a GS coupled receptor is activated, that stimulates the production of cyclic AMP, which increases conductance through cyclic nucleotide gated cation channels. That's what's shown on the left. On the right, GI coupled decreases cyclic AMP and decreases cation influx. Thus the neuron is inhibited. Norepinephrine, again, is created in a subset of neurons, just as it was in Lecture 5. What it's going to do is dependent on the receptor. And here's the deal. We got all three classes, just like glutamate receptors. Kind of easy to remember. Notice I say kinda. Beta. There's alpha and beta. Beta is always GS coupled. No ifs, ands, or buts. Now there's beta 1, beta 2, beta 3 subunits, and these are just located on different types of cells. If you're a smooth muscle, they inhibit you. If you're not, they stimulate you. Then we've got alpha, which unfortunately has an alpha 1 and an alpha 2, and they don't use the same G protein. Bah! Alpha 1, GQ, therefore it is always stimulating. Whether you're a smooth muscle cell or not, this will stimulate you. Alpha 2, GI. So it's inhibitory to everything except smooth muscle. That means that norepinephrine can do anything under the sun because it has all three types of G proteins. Well, it can do anything except directly activate an ion channel. Again, there are no ionotropic receptors for norepinephrine. So the catecholamines, dopamine, norepinephrine, epinephrine, have no ionotropic receptors. Serotonin does have an ionotropic receptor, but it's rarely used. Mostly, we're dealing with metabotropic receptors for serotonin. Again, made in the RAF-A nuclei, and the receptor is going to be either the 5-HT1, 5-HT2, or 5-HT3 type. 5-HT1 is GI coupled, 5-HT2 GQ, so we can inhibit, we can excite, and then 5-HT3 cation channels. In case you're worried about having to remember all these GIs and GQs and all that jazz, I'll put in parentheses on the exam. If I say it's a 5-HT2 receptor, I'll remind you GQ coupled. So know what GS, GI, and GQ mean, but you can sort of, maybe a little bit, forget the specifics of the receptors. Unless I forget to put it in the parentheses, then you're screwed. I think this might be the last neurotransmitter here. Histamine is there to promote wakefulness. That's largely what it does. It's made in the tuberomammillary nucleus of the hypothalamus, and these histaminergic neurons project widely throughout the central nervous system, waking up all the neurons that it talks to, except for itself. Histamine will turn off the histaminergic neurons because they have GI coupled H3 receptors. But everything else has either H1 or H2, and that's GQ coupled, maybe a little GS with the H2. Okay, so we got this wide distribution of histaminergic projections. What's it doing? Well, whether you're a cortical neuron or a neuron here in the substantia nigra, histamine stimulates you. Okay, look on the top. There's your control firing rate. It's just pacemaking away. Apply a little histamine. Notice the rate of pacemaking has increased. Maybe 30-fold. That's what I get from looking at plot B anyway. All the little dots there, you'll notice, we're just following over time on the x-axis, and the y-axis is showing us the firing rate. Normalized to baseline. So before we put any histamine on, take a look at it and call it 100%. That's its typical firing rate. Put on some histamine, it increases about 30%. Put on a drug that blocks the H1 receptor, and we're back down to 100%. And this just tells us that we have H1 histamine receptors on our SNR neurons. Now, if I were to tell you that there are H1 GQ coupled histamine receptors, you should know that excites a neuron. If I tell you that there's H3 GI coupled histamine receptors, you should know that's going to inhibit the neuron. Now, if there's anything else you'd like to know about further, make sure you put it in the questions box so I can field that in class. I'll see you later.